Hello, students. Very good afternoon. So today we have to continue from where we left from the last class. So before we head on to that, so let me launch you a technical poll. Okay, please give me a confirmation if my audio and video is clear. Okay. Yes, perfect. Thank you. So students from YouTube, if you have any problems concerning to technical issues, please feel free to type your concerns in the chat box. So let's get going in today's session. So we have some exciting topics. Okay. So let's see what we have in the box today. Yeah. So as usual, before we start, we have few things to do. That is attend a quiz session, right? So here is your first question for the day. A bipolar neuron has, okay, your options are 2 dendrites and 1 axon, 2 axons and 1 dendrite, 1 dendrite and 1 axon, 2 axons and 2 dendrites. Okay. So I will launch a poll, MCQ poll. So I want you all to answer as quick as possible because we discussed this in the last class. So it would make me more confident. Very nice. Hold on. Yes. Perfect. Perfect. Okay, let's see if we have what answers we have in on YouTube. Yeah. This is called quickness. Okay. Direct question. It's easy because you read about it. Correct. So let me end the quiz right now because let me not keep a suspense. Your answer is absolutely right. Okay. I'll end the poll. Yes. What is a bipolar neuron? A bipolar neuron is the one that has 1 axon and 1 dendrite, and that is option C. And your answer is absolutely right. Okay. So if you talk about unipolar, okay, and multipolar axons, correct? So you would already know the guess, like if it has many dendrites and if it has 1 axon, that is called as multipolar. If it has only 1 axon and 1 dendrite, it is called as bipolar. And unipolar, it will be only 1 axon without a dendrite. Correct, right? So let's go for the next question. The nerves carrying the impulses to the CNS are known as, okay. So again, I will launch a poll. Let's make it quick. Even this is a direct question. The nerve carrying the impulses to the CNS are known as motor, efferent, afferent, mixed. Okay, we already have an answer. Let's check it out. Yeah, nice. This feels good. Good interaction, good sessions, right? Nice. Come on, we have only 10 seconds more. We need more answers. All right, I'm gonna end the poll right now. So let's see what's your right answer. So you have answered option C, that is afferent neurons, and it is absolutely right. Okay, hold on one second. Where is my marker? Yeah, here it is. Yeah, afferent. What's happening? Oh, sorry, kids, 1 second. Come on. Yeah, sorry. Yeah, afferent neurons. So what are these afferent neurons? For example, if this is my hand, if not this is my hand, okay, let us say that we have hand. So the stimulus happens here somewhere, and the stimulus is carried from the site of occurrence all the way till your spinal cord. Okay, that is your CNS. So this relay of information from the site to the CNS is done by afferent neurons, whereas from the CNS back to your original position where the stimulus had occurred. That is called as efferent neurons. Okay, so we already know about it. Fine, people. So we will go a little bit backward from yesterday's class and then continue on with what we have to learn today. So we were talking about types of axon. A type of axons. We studied about multipolar, unipolar, okay, and bipolar neurons. So here we classify neurons based on types of axons. Correct. So we discussed this already. The first type is, one second. Yeah. The first type is myelinated axons. So what are myelinated axons? So these are the axons which are enveloped by Schwann cells, right? So they form, these Schwann cells are responsible for secretion of myelin around them. So myelin is basically providing a type of an insulation to your neurons, okay? So usually what happens is when you see the structure of a neuron, okay, like for example here, right? So you can see here, the neuron is not continuously coated by your myelin sheath. There are gaps in between. Okay. So these gaps where the myelin sheath is absent, that is called as, nodes of Ranvier. Okay. That is called as nodes of Ranvier. Right. So where are these neurons, myelinated neurons found? They are found in spinal nerves as well as cranial nerves. Right. So because of the presence of myelin, so these fibers or these nerve fibers appear white. So that is why they're known as white matter, right? So if they don't have myelin sheath, they start appearing gray. So that is why they're called as gray matter. Okay. So again, when it comes to another type of an axon, that is non-myelinated axon. So in non-myelinated axon, the Schwann cells are present, but they do not secrete any myelin sheath, or that means myelin sheath is absent. So they appear gray. As I said, they appear gray because of the absence of myelin sheath. So where are they found? They are found in autonomous as well as somatic neural system. Okay. So let's get on. Generation and conduction of nerve impulse. So this is very, very beautiful topic to understand how our neurons function. How does the carrying of information or the generation of this information takes place? Correct. It's such a complex, thing to even comprehend. You can't even just believe how beautifully designed it is, just by a very small reception, so much of information that we try to process in our brain, isn't it? So let us see how this even how does even the information originate in the first place. Okay, after the stimulation, after the stimulus is has occurred. Okay, I'm sorry about it. Yeah. So when we talk about neurons, as I said, neurons are the fundamental units, okay, of your nerve system. So when you see your neurons, neurons are excitable cells, right? So when we talk about excitations, their polarity can be changed, okay? So since they're excitable cells, and neurons are always in a polarized state, okay? This statement is very, very, very important. So when do you say that a neuron is polarized? Okay, for example, so let us say that I'll take an example of this neuron. Okay, so this is a neuron, correct? No, so outside of the neuron there is, okay, there is some area, correct? No, so this is the outer area of the neuron. So here is the area within the neuron. Okay, so if the outer area is more, okay, positively charged, okay, and the inner area of the neuron, that is within the cell, okay, if the charge is negative, okay, if the charge is negative within the cell, okay, compared to that of the outside it should be positive, and within the cell it should be negative. So at that stage we call, at that point we tell that our neurons are polarized. Is it clear? So when we talk about excitability of the neurons and the polarized state of neurons, so that is possible. See, the maintenance of this charge or the gradients of this charge is possible only by exchange of ions, correct? No, so here we don't have any, like switches. So the switches that we have that can allow the conduction of your charges, so those charges, the conduction of these charges or the passage of these charges occurs through the movement of ions, correct? No, so for the, if the movement of the ions have to occur, so what are ions? Ions are charged particles, isn't it? So for these ions to move across, it can happen only through ion channels that are present on the plasma membrane of your nerves, which is called as neural membrane. Is it clear? So neurons are excitable cells, right? So they can be excited, their polarity can be changed. Usually they're polarized state. Okay, they're in a polarized state. So what do we call, why do we call that neurons are polarized? I said you, when there is a charge difference, inside the cell it is negatively charged, outside the cell it is positively charged. So that is why, that is when we call the neuron as polarized. Correct, right? So that polarity is maintained. Okay, that polarized state is maintained by the exchange of ions, which are done through ion channels. Is it clear? Gates. Okay, ion gates you can tell. Next, how does the impulse conduction and transmission takes place? So for the impulse to even get transmitted, it has to happen in three important steps. So the first step for the transmission of the impulse is, first one is maintenance of resting membrane potential. So that is primarily important. So once the neuron is at resting potential, that means when the neuron is not reacting. For example, some of your body parts where there is no stimulus, or when neurons are relaxed, okay? So there is no conduction or information exchange that is happening, okay? So when this is happening, that is your neurons are at rest, or we can tell that they have a resting membrane potential. Once they are at the resting membrane potential, next phase is for the creation of an action potential. So once the action potential is created, now this action potential is converted into a neural impulse, and this impulse is propagated through action potential. Okay, so let us study these stages in very detailed manner. Okay, so for the first phase, okay, first thing that where we have to start is, what is the state of your neuron when is it, when nothing is happening, okay, when there is no nerve conduction, okay, or no impulse conduction is happening. At that phase, what is the state of your neuron, that we will see. Okay, so that phase where there is no impulse is happening, or there is no conduction of signals, at that phase we call it as resting potential. Okay, let's see what is the meaning of it. Okay, so as I said, when you talk about your neural membrane, that is a plasma membrane of your neuron, it is made up of various selectively permeable ionic channels. Okay, it is made up of various selectively permeable ionic channels. When you talk about resting neurons, right, when the neuron is at rest, now what happens is you have to consider two important areas here. One is, you can see here, in intracellular side, that is within the cell, okay, and extracellular side, that is outside of the cell. Is it clear, people? So there are the selectively permeable ion gates, isn't it? So when they are at rest, okay, now what they're doing is, when they're at rest, the potassium ions, you can see here, right, the potassium ions are very easily permeable. Okay, they are easily permeable into the cell. Okay, they come from outside to the inside of the cell very easily when the neuron is at rest. Is it clear? Fine. So what is not permitted? The sodium ions are strictly not permitted into the cell when it is at the resting state. Okay, so let us put it in a right statement. Whenever the neuron is at resting potential, okay, where there is no conduction of an impulse, okay, the permeability of potassium ions is more, okay, from outside to inside of the cell, whereas it is completely impermeable to sodium ions. All right. So now it's not always this balance that exists. So it has to keep on maintaining the balance. Okay. The balance is maintained by exchange of ions. Okay. Because of the selectively permeable nature of your plasma membrane of neurons. Now, what happens? Some of the negatively charged, one second. All right. Good afternoon. Okay. So when we talk about sorry, yeah, when we talk about permeability, right? Well, as I was saying, when we talk about the selective permeability of our neuron, correct? No, so they are allowing our potassium ions to flow in, and our sodium ions are strictly not permeable. And I also told you that this state of this balance does not exist all the time, right? So how does it maintain the balance at times? Whenever there is a difference in this charge balance, okay, that balance is maintained by the exchange of sodium and potassium ions. So how many sodium and potassium ions are exchanged? So for every, okay, for every 2 potassium ions, okay, 3 sodium ions are exchanged. Is it clear? For every 2 potassium ions, 3 sodium ions are exchanged. Is it clear? So to maintain the concentration gradient, 1 second, people. All right. Um, so I'm sorry, I have to divert a little bit. Um, Jamie Mahi, they asked me to introduce myself. I'm Vinay Anand. I'm a subject matter expert at Avanti. Okay, I teach biology. Okay, obvious, right? So that should be helpful. Sorry, kids. So let's move on with our discussion. All right. So let me get back. Let me get back because my flow is a little bit disrupted. Yeah. So as I was saying, when we talk about the exchange of ions, so the sodium ions as well as the negatively charged proteins are strictly impermeable, correct? So as well as to maintain this balance for every 2 potassium ions, 3 sodium ions are exchanged. That means 2 potassium ions are pulled inside and 3 sodium ions are moved outside. So that is how this balance is maintained, right? So when we talk about the fluid outside, that is we call it as interaxon fluid. Okay, they contain what? They contain very low concentration of sodium. Correct? No, when we talk about the extracellular side, there is very low concentration of sodium ion. You can see count the number of sodium to the number of potassium, isn't it? So the potassium ions are very low. Okay, whereas the sodium ions are very high on the outside. Okay, when you look at the inside, the number of potassium ions are higher compared to that of the sodium ions. Is it clear? You have to know which ion is higher, which ion is lower. Is it clear? So that difference in the sodium and potassium ions, right, number of sodium and potassium ions, it will create a concentration gradient. Okay. So this concentration gradient will make your inside of the cell what charged? Is it negatively charged or positively charged? What did I tell about the resting potential? Right? So you keep that in mind, we'll come across that, correct? No? So that concentration gradient is created across your resting membrane. Is it clear? Fine. So now, as I said, the exchange of sodium and potassium ions takes place across the membrane to maintain this gradient. Okay. So that is possible by the sodium potassium pump. Okay. So what did I tell? To maintain this balance for every 3 sodium ions, right, are moved outward for every 2 potassium ions to be moved inward of the cell. You can see here the direction of the potassium ions and the direction of the sodium ions. Correct? No? So now what will happen? The outer surface becomes more positively charged and the inner surface becomes more negatively charged. Remember this, in the resting state, the inside of the cell is more negative, whereas the outside of the cell is more positive. Is it clear? Now, when you talk about the resting potential, this potential difference, right? So this, the concentration gradient of ions is different. And you can see that there is positive charge and negative charge difference from the inside to the outside of the cell, correct? No? So this difference will create resting membrane potential. So this is called as resting potential. Is it clear? Okay. So if you understand resting potential, it will be very easier for you. Okay. I will summarize very quickly. So what is happening at the resting membrane resting potential? Your neuron is not conducting any impulse. So when it is not conducting any impulse, the ion channels are more permeable to the potassium ion. See, for example, if this is your nerve cell, okay, I'm sorry. Okay, I have to write this. Okay, okay, this is your nerve cell, for example. Okay, now what's happening? It is allowing more potassium ions and it is not at all allowing any of the sodium ions. Okay, sodium ions are strictly not permitted into the cell. Okay, when the cell is at the rest. Along with the sodium, even the negatively charged proteins are not at all allowed. And who does this job? Ion channels does this job. Clear? No? So now what is happening? So this gradient, the more number of positive, see, we have potassium ion concentration is more, as well as there are positively charged proteins. Sorry, yeah, positively charged proteins are there. So now what will happen? within your, okay, within the cell, and if you examine to the outside of the cell, there is a charge difference, correct? No? So this change in charge, so what change in the charge? The outer surface is positively charged, whereas the inner surface is negatively charged. You remember this. So this difference in the charge will create a potential. So this we call it as a resting potential. Clear? So now what will happen with the action potential and what is called as an action potential? So action potential begins with a stimulus. Okay. So for example, it might be touch. Okay. It might be a sight, or it might be audio, auditory, or it might be any of the receptions. Okay. The change in temperature or any of the stimulus that is around you or within you is felt from that localized area. For example, if I pinch myself, so I have to start from here, correct, right? From there, what is happening? Thus, at the site where the stimulus is induced, okay, now the membrane, okay, where the stimulus is generated, immediately these ion channels at the site of the stimulus, they become more permeable for the sodium ions. Is it clear? So once they become more permeable for the sodium ions, so this will cause rapid influx. Rapid influx means movement of sodium ions is happening from the outside of the cell to the inside of the cell. So once the sodium ions keep moving with, towards the inside of the cell, okay, now what will happen? Polarity will shift. Okay. So how the polarity will shift? Okay, gradually, when you look back, what is happening here? Your outer membrane is positively charged, whereas the inner is negatively charged, isn't it? Along with your sodium ions, some positively charged proteins also come inside your cell membrane, okay, within the cell. Okay. So along with sodium, the positively charged proteins also start moving it in the cell. So now the negatively charged cytoplasm will start become positively charged. Okay. So this reversal in the polarity is called as depolarization. Is it clear? Polarized, depolarized. Correct? No. So now it starts moving from outside will become more negative and inner side is more positive. Okay. Is it ulta? Okay. It's becoming reverse. Is it clear? So that is what action potential does. What is action potential doing? Once the stimulus is created, it will induce these ion channels to allow the permeability of sodium ions from outside to inside, along with some of the proteins which are positively charged. As the rush of the sodium ions and positive proteins move into the cell, now the reversal of polarity takes place. From outside will become negative, inner side will become more positive. And this process is called as depolarization. Okay. Now, what is happening here when we talk about electric potential? Okay. So what is electric potential difference? Now you can see that there is a difference in charge. If I take that, okay, this is the point of the stimuli creation. Okay. Stimulus is generated. You can see that only to a certain area there is a rush of the sodium ions and the positively charged proteins, not throughout the axon or not throughout the length of the cell. Okay. So here there is one type of charge. You can see here it is positively charged within the cell. As you move away from the site of the stimulus within the same cell, you can see that there is negative charge. You can see the difference is there, no? So this is called as electric potential difference. So when the electric potential difference across the membrane is created during the depolarization, so that is called as action potential. So what is an action potential? An electric potential difference across the membrane during depolarization. It is called as action potential. All right, people. Fine. Now, next we have to talk about how the signals get propagated. Okay, through after your the impulse is created. Okay, so once you have an influx of your sodium ions and positively charged proteins into the cell, now what is happening rapidly? Okay, let us say that this is point A and this is the point B. Okay, now point A, the stimuli happened. Okay, as the point A stimuli happened, the sodium ion channels opened and it starts flowing inward. Correct? No. Now what is happening? As the inflow of the sodium ion starts taking place, here what happens down the lane, okay, or down the cell or down the axon, now more and more of these ion channels keep opening, okay, in a series fashion, okay, like one by one, like gates, they keep happening. Now that will result in, okay, that will result in propagation of action potential. That means the rest of the cell, as you move from point A to point B, as the influx of your sodium and proteins keep happening along the cell length. Now, what is happening? The inner side of the cell along the length will keep turning positive. Okay. So as it turns positive, now what is happening? The impulse is propagated. Is it clear? Now, what's happening? Now, if you see, okay, if you see, as it keeps on moving in one direction from point A to point B, what is happening is it is very short-lived. Okay, action potential is very, very short-lived. As it moves or as it propagates from point A to point B along the axon, on the other hand, what is happening? As it moves along, now the ion gates channel gets reversed. Okay, it starts becoming repolarized. Repolarized in the sense, you remember, for every two potassium ions, three sodium ions are exchanged. That process keeps happening on the other end. Like see, this is the beginning, this is the later phase, and see the post phase of the impulse transmission. So you can see here, as the ion gates reverse their condition, here the repolarization starts taking place. Okay, again from B to A, the repolarization starts occurring. Okay, so that is how the propagation of the impulses takes place from point A to point B, and how after the conduction of the impulse, now your point A will start becoming to its natural state. It will come back to its natural state where it is negatively charged. So you have to remember one thing: whenever an impulse is created, people, whenever an impulse is created, now what is happening here? When an impulse is created, initially at the site of the stimuli, okay, the neuron which is negatively charged will become negative, will become positively charged. Okay, as the impulse is transmitted, now at the point where the action potential was generated, that will turn back to negative again. Okay, and this process is very, very quick. Remember that your neurons are way too fast. Because I'm talking so much, that means my neurons should be faster, right? So you know the capacity of your brain already. Okay, so I hope you understood. If you have any doubts, you can just put it. Okay, fine. Now this sequence is repeated along the axon, and the impulse is generated, okay, in a cyclic manner. Okay, from point A to B, B to A to B, B to A. This keeps on happening. Is it clear? So as I said, the permeability of sodium into the cell is extremely short-lived. Okay, that is why your cell will repolarize on a very rapid phase. Okay, so what is happening in repolarization? The potassium ions will diffuse outside the membrane and restores the membrane potential. Remember, for repolarization, what is happening? The potassium is not coming inside, the potassium is going outside. So as the potassium goes outside, now the more positive concentration was there within the cell, isn't it? It will start becoming negative at a quicker phase by the exchange of potassium ions. Okay, fine. Here is your question. During the transmission of nerve impulse through a nerve fiber, the potential on the inner side of the plasma membrane has which type of electric charge? Your options are: first positive, then negative, and continue to be positive; first negative, then positive, and continue to be positive; first positive, then negative, and again back to positive; first negative, then positive, and again back to negative. Okay, so I will launch a poll, student. Let's see if we get the right answer. Then that means you have understood this nerve impulse, okay, and transmission. By the meantime, I'll check on our students on YouTube. What's happening? Okay, students from YouTube, please feel free to type your answers on the chat box. Okay, let's make it more interactive. Okay, don't worry about the time lag. It's all right. Okay. Come on, students, make it fast. We just discussed. We have 10 more seconds. All right, let's end the poll. We have so much to discuss today. Okay, I'll end the poll. So you answered D, that is first negative, then positive, and again back to negative, right? So justification for your answer is here. First negative, then positive, and again negative. Correct now? So here is your option. That is option D is correct. Okay. So how does the transmission of this impulse takes place? So when we talk about the transmission of the impulse, so one neuron we saw what is happening in one neuron, how the action potential will help in propagation of the impulse from point A to point B we saw. So we have to understand how the signal is transmitted from one neuron to another neuron. So the transfer of signal from one neuron to another neuron happens at the point where you have two neurons coming together. So this point we call it as a junction. So we have a name for this junction, and that junction is called a synapse. Okay. Synapse is a functional junction between two neurons where the exchange of this impulse takes place. Okay. So there are two types of synapse that we can see. One is an electrical synapse, other one is a chemical synapse. Okay. When we talk about electrical synapse is very rare in our body, and it is so quick. Okay, it is so quick. And the second one is for an electrical impulse to to be transmitted from one neuron to another neuron, then the impulse that is traveling from a pre-synaptic to post-synaptic neuron, that is from one neuron. This is pre. Okay, let us say that this is pre and this is post. Okay, so for it to travel from here to here, it cannot do so through electrical synapse if the gap is too high. Correct? No? So like how your circuit has to be closed, similarly, even in neurons, it has to be as close as possible. So when they are in close proximity, okay, if the pre and the post neurons are in close proximity, then the electrical simulation or the electrical transmission of impulses will take place. As I said, they're very, very fast than chemical synapses, and they're very rare in human beings. When it comes to chemical synapses, so we know that we have our synaptic bulb, correct? No, or we also call it as synaptic knob. Okay, when we talk about the synaptic knob, which is present in the presynaptic nerve. Presynaptic nerve is the one which is actually giving the information. Okay, so when we talk about the end of this nerve, which is a presynaptic knob, what you can see is at the knob there are vesicles. Okay, so these vesicles, they contain specialized proteins or a specialized signaling molecules called as neurotransmitters. Okay, for example, acetylcholine or adrenaline are the examples for neurotransmitters. Okay, so if you see from pre to post synaptic nerve, there is a fluid that is filled between these two, which we call it as synaptic cleft. Okay, the junction or the gap that is between the pre and the post synaptic neuron is called as synaptic cleft. Okay, so when a signal is generated, okay, these vesicles come closer to the plasma membrane of your neuron. Okay, they will disassociate. Okay, or these vesicles will dissolve into the what do you tell the plasma membrane of the neuron, and then they start releasing these neurotransmitters. So these neurotransmitters they don't go and bind to the other neuron very randomly. Okay, for them to even cause an action, they have to go and bind to the specific receptors. Receptors are like lock and key, right? So if they have to induce a specific response, they have to go and bind to the specific receptors. Only when they bind to the specific receptors, a specific type of an impulse is generated. Okay, so when we talk about chemical synapse, they are very elaborate. Okay, they are very, very accurate when we compare it with electrical synapse. Okay, so what is happening in chemical synapse? In chemical synapse, we have impulse that is generated will reach the axon terminal. Now the synaptic vesicles will go and bind to the plasma membrane and dissolve, and they release neurotransmitters. So these neurotransmitters dissolve across the synaptic cleft or diffuse across the synaptic cleft, and then they go and bind to the receptors on the postsynaptic membrane. As they bind to the postsynaptic membrane, now the ion channels open, and again the action potential is generated, and your new nerve impulse will start continue. Okay, so this is about chemical synapse. All right, people. So the next part we have to discuss is the central neural system. So when we talk about the central nerve system, basically, the central nerve system includes brain and spinal cord. Okay, so let's talk about our brain. So it's unimaginable, and it is even hard to comprehend the complexity and the design and the nature and the capabilities of your brain. Not just your brain. If you take any organisms, take for example king cobra or tiger or any of these, like a eagle, okay, or a fish, all of them have a very complicated okay, nerve, complicated brain. They have extraordinary capacities. For example, elephants, they are very, very intelligent. Okay, orcas, whales. Similarly, human beings, we are so calculus in nature. We understand algorithms, we understand computing languages, right? We do math, we learn science. You listen to boring lectures like this. Okay, so many things we do, correct? No, so our brain, when we just look at the human perspective in thinking and like speech or emotional expressions, okay, being angry, being focused, or being sad, or being depressed, or feeling of missing, or feeling of love, empathy, all these are humanly behavior. When we look at the humanly behavior itself, it is so complex. But look at the capacity of our brain. Our brain not just gives you a potential to sense these feelings, okay, like emotions. It also gives you an incredible capacity of memory. It will control your involuntary organs. It will also give you a control over voluntary organs. It will give you, if I have to move my hand, I can control my hand movements, correct? No, so that is all this information is processed in the brain. Whereas when we talk about involuntary organs like stomach, liver, kidneys, okay, imagine that I have to think about breathing every single minute or every single time I have to take breath. Would I be breathing? So we are so lazy, we would forget breathing, correct? No. So similarly, if you talk about the brain's function, it is quite elaborate, as I say. Okay, control of involuntary organs, control of voluntary organs. Okay, giving you biological rhythms called as circadian rhythms, where you have a 24-hour format. We will feel, okay, this is night, I feel sleepy. Okay, your dream state. Okay, and then you have your deep sleep state. Then you wake up again and you feel energized. Correct? No, and to maintenance of your hormonal balance. Okay, maintenance of your body temperature, maintenance of your blood pressure. Okay, and ability to feel thirst, ability to feel hunger. The list goes on and on and on. Correct? No, so that is brain. So we are gifted with a beautiful brain. Okay, through evolution. Whoever designed it, okay, he is a masterpiece. Okay, he's the greatest engineer of all time. So, anyways, so when we talk about brain, so brain is basically present in the cranium or cranial cavity, which you call it as the skull. Okay, within the skull, it is present in the cranial cavity. So if we just look at the, our assumption is this is the skull and just below the skull is our brain. No, absolutely not, because even our evolution knows that this is a very vital organ and it needs extra protection. And that extra protection comes just below the skull in the form of 3 layers. Okay, so these 3 layers are basically connective tissue membranes, which are called as cranial meninges. Okay, which are called as cranial meninges. What are these 3 cranial meninges? We have the outermost layer. You can see here. You can see here. This is the skull. Here the skull ends. From here to here, okay, is your dura mater. From here to here is your arachnoid matter. From here to here is your pia mater. Okay, let's see them. The 3 layers of this meninges are the outermost dura mater, the middle arachnoid matter, and the inner pia mater. Okay, is it clear, people? So fine. So when we talk about the there is a space, okay, called as subarachnoid space. Okay, so this space is between the pia mater and the arachnoid matter. You can see here. Here is the pia mater, and here is the arachnoid matter, and here is the space. Okay, so this space is called as subarachnoid space. So this subarachnoid space is filled by a specialized fluid called as cerebrospinal fluid. It has a lot of role to play. It's also called as CSF. Okay. So when you talk about the ventricles of your brain, they are also filled with cerebrospinal fluid, not really the blood. Okay. Chalo. So now to talk about the further aspects of this brain, I wish to take in the next class because we have only 1 minute left. we can end the session now. If you have any doubts, you can post it in the chat box. Okay. Thank you, students. All right then. So if you have doubts, please feel free to post your questions in the chat box in the coming session. The time is up. Probably it's time for the next class too. All right, students. Have a nice day. Take care. Bye bye.